Yeah, this is no hormone lecture. This is an energy lecture. Oh, it's great. It is just so delightful to get to be here with you. I remember doing IFMCP back in 2003. So I've been in the functional medicine space for over 20 years. And the amazing thing is just how insanely overwhelmed I was by learning all of this and going through. Any feelings like that going on? Can I hear an amen? And you know, that is as it should be. As we were having a great conversation before the talk, you can't shift a paradigm without reassessing reality. You can't shift a paradigm without getting new information and making new connections. And how important it is to recognize that you're creating a new system of how you think. This is a different way of looking at medicine. It's not necessarily new information. So many of the things you've heard are things you've known before, but instead we're really grappling with the fact that reality is complex and that humans are systems. And how do we, with a good ethic, a good mind, a good heart, go after understanding this complex, dynamic system that is a human? And as we do that, we have this great opportunity to learn about ourselves and to question how we've organized our information in the past. And systems always require learning new data, first of all. Like imagine, think of the system of language. You had to learn your letters, and then you had to learn how letters, and then that was data. And then you learned how they connected, and that was words. Now you had words, that's new data. And you learn how they connect. Those are sentences. And those sentences are new data. And you learn how they connect, and that's going into a story. And so the process of learning a new system is always going to be this never-ending process of learning new discrete data and then recognizing how it's connected together. Does that make sense? So I absolve you from any overwhelm you're experiencing right now. You are enough. What you know even now is enough. And wherever you are starting from, this is where you need to be. And you're going to take what you know, this amazing history you have, this amazing background you have, the biases and the knowledge and all that, you're going to put that together, and that's your data. And you're making new connections. And as soon as you make those new connections, you're going to have a new set of data and another set of questions. And that's why we really call functional medicine the medicine of why. We're always open to asking another question, open to being proven wrong, and opening to find a better model so we can find better help and better answers. Does that make sense? So anyway, I'm so thankful to get to be here with you. It is just a joy to get to participate in seeing what I see as the transformation of medicine across the world. And so this is our matrix. You are well familiar with it at this time. And we're going to be zoning in on the energy side. Now, when we say energy in functional medicine, it kind of sounds like, what's that mean? If we look at function of energy, it is to fuel and to enable everything that goes on. There's energy within any system. There's the hard parts of a system, and then the energy is what connects. The energy is what enables. And so the great metabolic enabler that we're going to talk about today is the mitochondria. The mitochondria is our source of energy that makes every cell in your body be able to do what that cell was designed to do. And if it doesn't have sufficient energy, we're literally not going to be able to fulfill that cell's life purpose. So we want to step back and not be, so any of you who had kind of like post-traumatic Krebs cycle disorder, right? Anyone, right? We're going to try and absolve, going to do a little EMDR on that. Huh? But we're going to go straight, first of all, in diving into the structure of the mitochondria. Because structure and function are always intimately linked. This slide is really all you need to know. Multiple sources of energy, be they fats, proteins, or carbohydrates, can get broken down in the body to create ATP, create the energy pool for all of the active transport components that are going on in life. So start with the fundamentals. What we're understanding is that every cell needs energy, and therefore our body has multiple backup systems in order to make certain that it can survive and make life. So what are mitochondria? So what the mitochondria and chloroplasts contain DNA that's distinct from nuclear DNA. These chloroplast genes are related to free-living cyanobacteria, mitochondrial to proteobacteria, and the invagination of these archaic bacteria created a symbiosis and basically a division of labor where the nuclear cell, the eukaryotic cell with its nuclear DNA, outsourced all the dirty work to the mitochondria. And the mitochondria said, whoa, we've got a lot of sensitive DNA and information here. Let's get rid of that and outsource that to the nuclear DNA where it's better protected. So the mitochondria that live inside of us, inside each cell, are bacterial-like as their origin would show us. So structure and function, structure, function, structure, function. So here we have the external membrane, which is highly permeable. It allows things to come through. And then we have an inner membrane, which is highly impermeable, does not allow especially hydrogen ions to come through, or many other compounds for that matter. And in between, we have the intermembranous space. That is essentially what's going to create the battery that is the charge differential that will drive the engine of making ATP. So, and then the inside is the matrix. I just love the fact that we have one of the best movies of all time is called The Matrix, right? I really always feel like I want to start this talk with, do you want the red pill or the blue pill? Because we're going to enter the matrix right now. So the mitochondrial structure, we've already talked about, outer phospholipid layer, intermembranous space, the inner membrane. The cristae are the massive amount of invaginations and evaginations that increase the surface area of the mitochondria, and which contain the electron transport chains. So the more surface area you have of the cristae, the more machines you have to be able to make ATP. More cristae, more energy production capacity. Damaged cristae, which means a damaged inner membrane, you have battery leak. You literally have leakage of the hydrogen ions. Inside the matrix, this is where we keep all of the mitochondrial DNA. Proteins, glutathione. About 15% of our body's glutathione is stored inside the matrix of the mitochondria because this is our furnace. Calcium is sequestered in this environment as a highly reactive molecule, and so the release of calcium starts so many different kinases up in cellular signaling. The mitochondria are exceedingly important cellular regulation outside of ATP production just because of how they sequester and control calcium. And then the NADH to NAD ratio helps us understand that NADH as being a potential energy, NAD more the spent energy molecule, and all this is abundant inside the matrix. So let's go into the cristae. And very large surface area improves efficiency. The cardiolipin is the major phospholipid in this area, and its structure actually allows this membrane to bend. Again, a structure-function relationship. Whenever we have a molecule, it's going to create a structure that will then create a function. Goes back to the system, right? Discrete data, connections, making a system. And so the more ATP demand that you have, the more cristae will form. The more cristae, the more citrate synthase you will develop. And citrate synthase, you'll find in the literature, is a good surrogate marker for how much of this membrane you have available. Often a surrogate marker for how much mitochondrial mass is present. If the cristae are healthy, you maintain the battery. If the cristae are damaged, you lose potential energy. And we're going to talk about a whole bunch of things that damage cristae, a whole bunch of components. What's in this mitochondrial matrix? The mitochondrial DNA is a very special DNA. It doesn't have as much protection as nuclear DNA. The inside, they have ribosomes for making the specific mitochondrial proteins, transfer RNAs, proteins, and there are over 1,500 proteins inside this matrix. And as we'll learn, we only have 37 genes of mitochondrial in the mitochondria. So the proteins that are inside the mitochondria, I love the fact that 30% we have no known function for yet. Keeps us really humble. And, but the transport proteins that actually pull molecules into the matrix are exceedingly important, and many dysfunctions that we see have to do with the proteins that diminish the ability to transport needed molecules from the cytoplasm into the mitochondria. So mitochondria are unique. They look different depending which cell they're in. If they're in a long cell, such as a peripheral nerve, they're a long, stringy thread. If instead they're in a hepatocyte, they'll be round to maximize the efficiency. There's no limits on the size shapes. They're highly mobile. They move around inside the cytoskeleton. They're constantly undergoing a process of fusion and fission, fusion and fission. We'll go into that in depth in the energy module, because it's a major way that we regulate so many functions in the body. So the disorders of mitochondrial dynamics really span every possible disease that there are. We've gone from the idea that we only have primary mitochondrial diseases, and those are the single nucleotide or multiple nucleotide catastrophic disease that often ends with early in life death. Those are the primary mitochondrial diseases. And increasingly, we recognize that there's a vast amount of secondary mitochondrial dysfunction. And that's what we're here today, is to talk about the mechanisms of understanding how does a mitochondria dysfunction. And you will recognize that the mitochondria is always in this symbiosis, this ongoing conversation with the inside of the cytoplasm, inside of the milieu of the cell. Mitochondrial DNA, we get them from our mother, vastly inherited. There's theoretically some that could come from the sperm, but that's really just an exception for winning a trivia contest. Has no meaningful. But we get our mitochondria from our mother. The ovum contains about 100,000 to 1 million mitochondrial DNA molecules. So here's a trick question for you. How many mitochondria, then, are in the ovum? So if you have 100,000 to 1 million mitochondrial DNA copies, how many mitochondria are in the ovum? 1. It's 1 mitochondria. It's like a lava lamp. Any of you have a lava lamp when you grew up? I totally love lava lamps now, right? Because they're like mitochondria. They come out and they bleb out in a little bit of fission, and then they fuse back together, and they fizz together, and they fuse and fizz. And that is this constant dynamic. So essentially you have 1 mitochondrial mass inside the cell. So counting numbers of mitochondria is what we do in an electron microscopy, because that's what we can see, these discrete entities. But they're constantly in movement. They are constantly changing, and they're constantly in flux. This is why what we do matters, because we can change mitochondrial mass. We can change mitochondrial function by how we give those new mitochondria a new milieu, a new macro and micro environment. So, mitochondrial DNA is circular. It's like a bacterial DNA, 37 genes. Nuclear DNA that controls the mitochondria, which we call the mitome, is about 1,000 genes. There's every form of genetic transmission in mitochondrial diseases exists. These mitochondrial DNA are highly susceptible to oxidative damage because they are not wrapped in histones as so many as the chromosomal DNA. So they have a much higher number of injuries on a per unit time basis than the nuclear DNA. And a very important repair capacity goes on with mitochondrial DNA. About 10% of your body weight is mitochondria. Certainly more if you have more lean body mass as far as percentage-wise. And they make and create your weight in ATP every day. Just sit on that for a second, right? I mean, you're making, using, recycling your body weight in ATP every day. And so a small amount of dysfunction makes a large difference in cellular capacity. And if the cells don't have the energy they need to do the work they do through their primary and preferred pathway, your cells and your body is really smart, and it turns on all of the compensatory systems to start enabling that cell to limp by. And if that's allowed to proceed, that's cellular dysfunction, which leads to tissue dysfunction, organ dysfunction, and eventually that dysfunction leads to disease. So this is looking at cause from an energetic standpoint. Mitochondria actually are located inside the cell depending upon where they're needed for energy. So in the renal tubular cells, they're right up against the distal portion of the renocyte. The mitochondria make up over 50% of the mitochondrial cytoplasm, and they're closely coupled to endoplasmic reticulum, because the endoplasmic reticulum also regulates calcium flux inside the cell. So mitochondria, we've already said that they make energy. They're important for biosynthesis, so they make molecules, and they're important for signaling. So energy, we talked about ATP. They maintain this ion gradient, which enables what we'll see the electron current to flow to make ATP. They make reactive oxidative species, which we'll learn are signaling molecules. They buffer this calcium ion flux. When mitochondria die and those fragments get into the surrounding cellular milieu, it's a great danger signal. Because if your mitochondria are dying, it's a very strong signal the cell needs to die. So it's a strong signaling molecule. And they have a large part in regulating cell growth, repair, senescence, and the ability of being able to grow and not grow. All right. Energy. So when you think mitochondria, why would the nuclear cell, why would the eukaryotic cell want to outsource its energy-making capacity into a subcellular architecture? Because the best way to make energy is with oxygen. And oxygen is so highly reactive, it tends to damage everything around. And so the way I explain mitochondrial function to my patients is I say, well, you probably drove here today in an internal combustion engine. It always used to be the case. Now there's more Teslas. So they still remember what an internal combustion engine is, though, because that's what a mitochondria is. And think of that engine. Inside flows oxygen and fuel, and then that gets burned. It gets triggered and consumed. And out comes a different form of energy, which maybe transported your vehicle here today. Out comes carbon dioxide, out comes water, and out comes soot and smoke. And the soot and smoke of the mitochondria are the oxidative radicals that we'll talk about more. And those oxidative radicals damage all the cellular architecture, all of the structure that enables the function of that subcellular structure. So this is why we want to outsource energy making to a mitochondria, because it's a dirty business. Dirty business. So the primary proton pumps that will actually take advantage of this electron gradient are complexes 1, 3, and 4. And increasingly, we can order labs that can measure the activity of these complexes in the body. And as we learn more about mitochondria and how they influence disease, it's fascinating to see that start to change. Fats, carbohydrates, and proteins. So this is an expanded slide of what I showed you before, that there are different mechanisms to bring energy through the cycle of breaking down macromolecules through pyruvate, finally onto acetyl-CoA, acetyl-CoA going into the Krebs cycle, the Krebs cycle making the energy intermediates that drive the electron transport chain, that make the proton gradient, that enable the ATP synthase to work. It's a beautiful, beautiful dance that occurs. Now, so there's 3 mechanisms we break down macromolecules: glycolysis, the citric acid cycle, and beta oxidation. So when we think of these mitochondrial bioenergetics, we're dependent on these intermediate molecules. NADH and FADH2 to transfer the energy that's being made inside the Krebs cycle to the electron transport chain. Here we see this a nice graphic form as acetyl-CoA as a centerpiece of how we take these large molecules into 2 carbon units and shove them down the pathway. Now, glycolysis, this is a very ancient pathway. It's just going to break a sugar into 2. If you break a sugar into 2, you get 2 NADHs and 2 ATP. Very simple. Pyruvate is actively transported into the matrix to enable aerobic respiration. So pyruvate is then going to interact with oxygen, and it's going to make many more ATP possible. When mitochondrial metabolism is inhibited, you see this pyruvate converted into lactate by lactate dehydrogenase, and that then regenerates NADH. Do any of you measure lactate dehydrogenase? LDH? Right? LDH, it's a very common standard lab. Did you know you're measuring mitochondrial function? So you're literally, when you see an elevated LDH, yes, that could be a cellular injury, but it's more likely that you're actually having a cellular energy demand issue. And the cell is saying, we need to upregulate the production of LDH so we can convert more lactate into pyruvate because we're not able to get energy from other means. So rethinking what molecules mean is actually part of this process. Maybe those aren't liver enzymes, maybe those are mitochondrial markers. So glucose and fatty acids both, so glucose breaks down to pyruvate, and pyruvate can go to lactate. Fatty acids also break down into a 2-carbon unit and join acetyl-CoA, and then acetyl-CoA goes on and can enter into aerobic metabolism. Acetyl-CoA is this substrate for the Krebs cycle, or the tricarboxylic acid cycle. And it's important to balance out between, it's there so that there's one engine to be able to utilize energy sources from either fats, proteins, or carbohydrates. The Tca cycle is really this final catabolic pathway for all nutrients, and it produces NADH, FADH, and GTP. This is a great, if you print out one slide, print this out. This has all the necessary components of where energy is coming from and where it's going to. And I'll point to this slide here. Again, ta-da. So we see glucose coming in the top, fatty acids coming in the top. And they are being able to be broken down, glucose into pyruvate, pyruvate into acetyl-CoA, and acetyl-CoA enters into the Krebs cycle. Whee! Can everybody say wee? Because that's how I feel like this happens. It's definitely a wee. Wee! And makes FADH2 and NADH. And then these are the energy intermediates that enter into the electron transport chain, drive protons across. Those protons going across come back through to enable the production of ATP. But if you're fatty acids, instead, it gets broken down by beta oxidation. And I like how beta oxidation is just this big swirl. Because if you really want to give yourself a migraine, look through actually how beta oxidation works. It's much better as a swirl, let me tell you. And this can enter directly into the electron transport chain through beta oxidation. And that's really cool because there's a bypass route to bring energy in straight through CoQ10 in to make the electron transport chain. So what is so important to recognize is energy is a system. It's not linear. This is why we can survive. There's all kinds of dysfunctions that occur, but our body is going to adapt. And sometimes those adaptations help us in the short term and harm us in the long term, ergo disease. Adaptation. So, now beta oxidation is this multi-step process. We are breaking down long-chain fats into ketone bodies, which are 2 carbon units. These ketone bodies go by several names: beta-hydroxybutyrate, acetoacetate, and acetone. And when we bring in high amounts of carbohydrates, what happens to the process of beta oxidation? it turns off. Carbohydrates are basically carbons that come with their own oxygen source. They're exceedingly efficient to run with, hot burning. The mitochondria do prefer to burn that glucose first. And so if glucose is present, it's going to turn off the breakdown process for long-chain fats. So when we start seeing this process occur, and you continue to bring in glucose, you have the buildup then of intracellular fats, ergo fatty liver disease once it gets to a macro stage. And these fats buildup goes hand in hand both as cause and effect of insulin resistance. So how can you address this? Fasting, starvation, low carbohydrate and high fat diets increase this hepatic beta oxidation, causing more ketogenesis, but also turning off glycolysis. So ketones are soluble in water. There's no protein carriers required. It's a direct energy source. The plasma levels of ketones go up with fasting and with the high fat and low carbohydrate diets. Ketones are really the preferred fuel for the cardiomyocytes as well as the renal cortex. That's kind of a cool thought, isn't it? So, many times when you're looking at a renal diet, they want to decrease the amount of protein in that diet. But the wrong compensation is necessarily to have a very high carbohydrate. More of those calories should be probably coming in as fats and beta oxidation enabled. So in thinking about what's the balance of going on, how do we help cellular function more? So ketones are used in the brain after crossing the blood-brain barrier, really proportionate to the concentration in blood, and they give energy when glucose supply is limited. Amino acids can also be used as fuel. They can be broken down, and about 10 to 15% of your daily energy creation comes from protein, comes from the breakdown of amino acids. And so they also become the precursors for gluconeogenesis when glucose supply is low. So if the body needs more glucose, it will break down muscle. And we know the breakdown of muscle becomes a condition called sarcopenia, literally lack of flesh, a very high risk marker for virtually every chronic condition that occurs. So here we see details how specific amino acids not only participate in the production of energy, but they also will substitute into the Krebs cycle. They will substitute into individual markers within the Krebs cycle and serve as a backup process. If the body can't make an intermediate in the Krebs cycle, it can break down protein to find amino acids to accomplish that task. But again, what has happened? The breakdown of muscle. And with less muscle, no good things happen. Krebs cycle, we see this diagrammatically, fats, carbohydrates, proteins, coming into acetyl-CoA, and then acetyl-CoA joins with oxalic acid. This becomes citric acid. And remember I said citric acid synthase is a good surrogate marker for the amount of mitochondrial mass. Citric acid becomes cis-aconic acid, isocitric acid, alpha-glutaric acid, finally to succinic acid, fumaric acid, malic acid, oxaloacidic acid, and then back to citric acid. It's possible to measure these intermediates, and by looking at the ratios of one to the other, think of this, you can start hypothesizing what is going on with function inside the cells. And I really like to use the word hypothesize, because remember, I've been talking system. Whenever anybody gets too reductionistic by making, this number is high, therefore this is going on, it's kind of the mark they don't know what they're talking about. Because the body has so many adaptive strategies, it could mean several things. But this enables us to think through and say, what are safe and effective ways that I could help move this person forward? So measurement of individual organic acids is one method of assessment. Now, this whole process leads us to oxidative metabolism. How do we burn molecules using oxygen to make NADH and FADH2? And And I think the best way to look at this is to, as we've talked through the process, is to see it in action. So selected pathways in mitochondrial metabolism. Does that help you set up to think about mitochondrial metabolism a little differently? Right? Why are we doing it? All of it's about making food into ATP, and it's going through this gauntlet to actually occur.